to the session. Oh, great. Thank you for recording. Yeah, so welcome everyone to the session on digital evolution and evolutionary applications. I'm really excited to see everyone's talks. I think we've got a really great lineup. Just as a reminder, talks are 15-minute total blocks. So ideally that should be 12 minutes of talking and 3 minutes of questions. I will cut you off after 15 minutes, so please try to stay within time. And without any further ado, let's hand it over to Chris Adami, who will be doing today's first talk.
All right, thank you, Emily. Let me do my sharing and hit play. Let me also start my timer. So here's the boring talk that I advertised, which is actually the title of the paper I'm talking about. And I realized I should have really, made it more dazzling by putting in something like this world-breaking, complete genotype-phenotype map, which maybe would have doubled the audience right then and there. So that's what I'm going to be talking about, genotype-phenotype maps. And so let me first tell you what they are. Generally speaking, this genotype-phenotype map, and I'm probably going to say GP map a lot, is a many-to-one map of genotypes to phenotypes. In a sense, you normally talk about this in terms of fitness landscape, which is in a sense a special case. Generally speaking, the genotype here will be a symbolic sequence, while the phenotype is a scalar or a real number. Actually, sometimes it can be even just like a structure, as we'll see. So typical examples are, of course, the map from DNA sequence to fitness of an organism, or the map from a protein sequence to the folding free energy delta G of that particular sequence. Or if the protein is a transcription factor, you might actually map the protein sequence to the binding affinity to a particular binding site that you can quantify. Or in the case of RNA sequences, people use this map a lot to understand what secondary structures all map, what sequences map into the same secondary structure. So these are all genotype-phenotype maps, and people use them for all kinds of different things. And the reason for that is that the GP map is really the interface between variation and selection, as you can imagine. If we would know this map precisely, in a sense, then the phenotype would not be a mystery anymore. And obviously, we would be much better at designing organisms. If selection acts directly or even indirectly on this phenotype variable, then this force, the selective force, will actually shape the probability distribution of sequences. It will do that slowly, of course, over evolutionary time, but certainly this distribution is going to be very important for evolutionary dynamics. In particular, if you just ask, you fix one phenotype, then there is a distribution of genotypes that's consistent with that phenotype. And what that distribution is, whether it's, for example, flat, or whether in fact it's, highly rugged with very, very high peaks and very low valleys, all of that stuff is going to shape evolutionary dynamics. Now, these GP maps actually also have a topology. And the reason for that is that this network of sequences, makes a map if you define the link between two sequences in terms of the mutational closeness under a particular operator. The operator, of course, usually is number of mutations. So typically the distance between two nodes in this network is just the number of mutations that separate them. And then, of course, each node with it carries a number, which is that the phenotype value that we're interested in. Typically, these GP maps have both local and global properties that people are interested in. A typical local property, for example, would be the number of neutral neighbors of one particular sequence within, this grander map. And a typical global property would be trying to understand the largest connected component of, all the sequences that you're interested in. So let me show you a very typical GP map analysis that comes from actually analyzing RNA secondary structures. It's a relationship between robustness and evolvability. So the genotype here is just this RNA sequence that you see here, and the phenotype would be a secondary structure. And if you define that, then you can define these neutral networks, which would be all of the genotypes that fold into this particular structure. And in fact, here you see such a map. So let me show you on the x-axis, you see actually the logarithm of this network size of a particular neutral network, connected network, divided by, in fact, the total number of sequences that are possible for this length L. So in a sense, this is here the density of structures that, of sequences that give rise to that structure. And on the y-axis, you see the fraction of neutral neighbors that each of these particular networks has. So each dot is one network, which is defined by the density and by the number of neutral neighbors. And this is a paper that came out, a few years ago. And one of the things I want to convince you is that this, analysis is utterly and completely trivial. And the reason why the authors didn't see that is because they didn't see that what they've written down here on the x-axis is actually an information-theoretic quantity. And I'll show you now how to understand that. So it is actually possible to relate the number of sequences that have the same phenotype to the information these sequences have about that phenotype. And the first one who wrote about it this way was actually Jack Szostak in this paper in 2003, where he introduced this concept of functional information. I'll show you how it's defined. I'm just going to read here what he wants to do with it. He basically says, a new measure of information called functional information is required to account for all possible sequences that could potentially carry out an equivalent biochemical function independent of the structure or mechanism used. In fact, in a sense, what you will see is that what was plotted on the x-axis of that plot before is in fact that measure. It turns out that Jack was actually inspired by my paper from 3 years earlier to define that. And in fact, he acknowledges that in the references, but that's now neither here nor there. Let me show you how it's defined. So first, Jack defines the fraction of sequences with functionality theta or beta, where theta could be anything like fitness, for example. So it would, for example, would be the fraction of sequences with fitness W or beta. And then he takes minus the logarithm of that fraction, which gives you an information. And that information is measured in units that are given by the base of the logarithm. What I'm going to do now is I'm going to apply it to sequences which are not RNA sequences, but there are in fact avidians, Avida self-replicators in particular. Since this is Beacon, I don't have to introduce what Avida is, and I won't have time for it. But I will tell you that these avidian sequences are written in terms of an alphabet with here 26 symbols, which are actually instructions to run on the computer. And these self-replicating programs in Avida are actually extremely rare. I wrote here a few of them, 3 of them actually. And it turns out that this is the smallest number of instructions that you can have in order to self-replicate in Avida. So generally speaking, the minimum length of Avidian self-replicators is length 8. And obviously now I can define a genotype-phenotype map by basically asking who is amongst you is a self-replicator. So that would be my minimum criterion. It turns out that we have previously, me meaning Nitash and I, mapped the landscape of length 8 self-replicators, and we found that there were exactly 941 self-replicators of this length out of a combined 209 billion sequences. Now that we know this number, we can actually calculate the information content of these self-replicators by the formula I showed you, and it turns out that it's about 5.9 mers. That means, in fact, because these are all 8 mers, that about 3/4 of these mers are information. Okay, so 6 mers out of 8 are information. Now what we did is we analyzed the complete length 9 landscape. And so you might think, well, going from length 8 to length 9, how bad can it be? And the answer is, well, now we actually have to look at 5.4 trillion sequences. And that means we're going to need a bigger computer. And it turns out that this ran for a full 3 months on ICER, while running, of course, all of this stuff, fully parallel. So I now show you the world's largest GP map to date. And when I say largest, it's because the previous record was length 20 RNA sequences, and this particular team, about 10 years ago, analyzed 1.1 trillion sequences, to which Nitash and I basically just said, hold my beer. So we found among these 5.4 trillion sequences exactly 36,171 replicators, which tells us that the information content is now 5.77 mers, which is actually significantly less than the information content of length 8 replicators. And you could ask, how is that possible? Turns out that the answer to that is sort of interesting, because the answer is that in order to compress information in a optimal way, you actually need extra space. So that extra mer that we provided, it actually allowed the organisms to compress the information more. But when I say organisms, of course, there's no evolution going on here. This is the fitness landscape before evolution. So in other words, what we can say is that if you allow for more extra space, then information can become compressed more optimally. So I'll show you now the largest cluster of sequences. It's about 94% of the sequences, so about 34,000. And you can actually look at the network structure, which I show you here. Now, this structure itself is enormously interesting, but unfortunately, I won't have time to talk about it. What you see, for example, is that there are 5 very weakly connected clusters, and each of these clusters consists of many tightly connected clusters, and each of these groups is an even more tightly connected cluster. So there's a hierarchy here that would be very interesting to analyze. Here's in fact the edge distribution of all of these sequences, and you see that it's highly varied. There is one sequence that actually has 74 nearest neighbors somewhere around here, and then there are groups that are all somehow have around the same number of nearest neighbors, which tells you that there's an enormous diversity of programs in here. Now, let's actually investigate how the information is encoded in these sequences. And the way we're going to do that is by defining the fraction of functional sequences within a radius n of a single replicator I. So basically, we'll do that. So, we'll go, okay, how many replicators are there within 1 or 2 or 3 steps, mutational steps, divided by how many there are at this step size. So this numerator here is the number of replicators k mutations away and then summed up till n. And the denominator here is the number of sequences k mutations away. So here's how that's going to look like. So I'm going to plot this distance out here and this log of this fraction over here, which essentially is the information. It's actually minus the information, but never mind that because I didn't put the minus sign here. So for example, if I go 1 step out, I basically get a certain amount of information that's in there. And as I go 2 steps out and so on. So if I go 9 steps out, of course, I have to reach this exact number of 5.77 mers, because by that time I have the entire landscape within my radius n. So if, in fact, from the sequence where I started, the information is accumulating in this stepwise manner, that means, in fact, that there is no epistasis between the instructions. But normally that's not the case. So here, for example, you would see where as I go 1 step out, 2 steps out, there's just a little bit of information accumulating. But of course, as I go on, at some point, again, you're going to have to reach that point. This is typically antagonistic epistasis. That means I have a very robust encoding. The opposite of that would be where after a few steps, I'm going to accumulate a lot of information very quickly, which then slows down. And that is, in fact, synergistic epistasis or very fragile encoding. And I should say also that there is a curve here, which is the most fragile encoding you can possibly do, which is, in fact, happening if there is only 1 single sequence. So there's never any neutral neighbors, and that's why this, of course, would end up here at 9 mers. So let me show you the actual data, what it looks like. And you see here is that red line of the most robust encoding. And you see that there are actually, these are the lines, by the way, of all 36,171 replicators. There's many that actually are very close to that most fragile encoding, but there are also some very more robust encodings. And in fact, I'll show you here sort of an aggregate. The red line is, in fact, that no epistasis line. The dashed line is the most robust sequence amongst all, as far as information is encoding is concerned. And down here is the least robust one. The red one, of course, no epistasis. And the black one is, how on average information is encoded. And I want you to take a look at this sequence here. This is one where information is so encoded in such a fragile manner that up to 4 instructions, there is not a single neutral neighbor within that sequence. In other words, it's a peak that is entirely by itself, within a radius of at least 4. But then as you go to 5, you start, accumulating, the rest of the information. So basically, my conclusions here are GP maps can be analyzed using information theory. And an information-theoretic analysis actually reveals much more than robust neutrality analysis, which in the light of the information theory turns out to be utterly trivial. The analysis quantifies both global information content and also how information is encoded in the sequence locally. And the analysis of the largest GP map to date shows a number of diverse information encoding strategies that will influence the evolvability of these sequences. And having said all this, I would like to thank Nitash, who actually ran the entire thing and did the analysis of them. So I'm grateful for that, and for ISER, of course, for giving us all that time, and for Beacon, who's always around. So with that, I am opening up for questions.
Thank you so much, Chris. That was fascinating. Unfortunately, we do not have time for questions. It was the full 15 minutes, but I am sure we're all super excited to ask you questions later because, yeah, super. I'm still thinking about that.
Thank you.
Awesome. So next up is my talk, and let me just share my screen. I'm gonna try and moderate myself. Okay. Okay, and starting my timer. Hi, everyone. I'm Emily. I just finished my first year as an assistant professor here at MSU, and I'm going to be talking about the evolutionary implications of phylogenetic diversity. And if you don't know what phylogenetic diversity is, don't worry, I'm about to explain. Just as a side note, all of the data I'm going to show here is coming from an evolutionary computation context. So where we're using evolution as a machine learning algorithm to better understand to solve computational problems. I think all of the insights I'm going to talk about here should also apply to non-computational systems, but obviously I haven't tested that. And so that's going to influence some of the way I'm talking about some of these things, just FYI. Okay, so I'm going to start with what I hope is a very uncontroversial point. Diversity is important for evolution. Variation is one of the 3 ingredients for evolution by natural selection. Anecdotally, in evolutionary computation, we frequently see that when evolutionary algorithms fail to solve a problem, the reason is that they run out of diversity. The entire population gets stuck on some bad solution. And there's a ton of evidence I could throw at you for this, but I'm going to ask you to trust me, diversity is important. But there is a problem with that statement, which is that diversity is not just one thing. If we imagine a population, maybe this population is shapes here, for instance, there are a lot of different ways we could measure its diversity. So a simple one is richness. So that's just the number of unique types of things that exist here. But that doesn't take into account, for instance, how evenly distributed the population is among those types. So if you imagine like a forest of oak trees, for instance, if there is one sycamore in there, that might not be as diverse as if the population were evenly balanced. And so if we care about that, we want to use a measurement of diversity that takes into account evenness, such as Shannon diversity. Now, both of those are examples of measurements of diversity that focus on the population as it exists right now. But often it's useful to incorporate more information into our concept of diversity. And so there's a large set of diversity metrics that fall into the category of phylogenetic diversity. So these are diversity metrics that pay attention to not just the diversity of things that exist, but the diversity of evolutionary history the population that we currently have represents as a whole. And there are a lot of different ways that we can measure this. They're all basically measurements that revolve around the topology of this phylogenetic tree, so this tree of ancestry of the population. And so I'm just going to refer to phylogenetic diversity as an abstract term. Know that it's not one thing either, but it is a useful class of diversity metrics. And so, there's a lot of reason that we might think that phylogenetic diversity would be more informative about what will happen in evolutionary scenarios than other types of diversity, since it is this notion of diversity that includes evolutionary history. But that's not a given. I think for a long time, people have kind of assumed that like phylogenetic diversity and other types of diversity contain a lot of overlapping information. And if we're going to start paying attention to phylogenetic diversity, it's important to first make sure that it kind of includes more information above and beyond other diversity metrics. This is particularly important in the context of evolutionary computation because people there historically have not even thought about phylogenetic diversity. So if we want to say they should start paying attention, we should provide evidence that it is, first of all, something different. So that's my first question. Are these types of diversity actually meaningfully different? So I was kind of first inspired to ask this question by these results I got a couple of years ago. So here I was doing evolutionary computation on a few different fitness landscapes. So each of these fitness landscapes are a different computational problem that we were trying to evolve solutions to. And I tried 4 different selection schemes on them. You don't need to worry at all for this talk about the specifics of these selection schemes. Just know that each one of them is a different way of selecting which individuals in the population get to reproduce. And so I measured phylogenetic diversity in these and saw they are going up. That's interesting. But the thing that I found really interesting is that when I looked at phenotypic diversity, in this case, this was Shannon diversity, but richness behaves similarly, it looks completely different. So that kind of implies that these things are telling us different information about what's happening in the population. And this is corroborated if we look even just within a specific type of selection scheme. So this is just one of those selection schemes, but I varied one of the parameters for it a little bit. And even here, the relationship between phenotypic diversity and phylogenetic diversity changes as I vary this parameter. So that suggests these are not the same thing. However, and there's also evidence from biology that suggests this preliminarily, but this evidence from biology kind of suggests that this is context dependent. And so before I kind of conclusively say that these are different things, I wanted to do a broader sampling of the space of possible fitness landscapes and selection schemes, basically ways of introducing different evolutionary dynamics into the system in order to see kind of what the broad relationships are. So this is going to be a lot of data and I'm going to go through it a little quickly. But basically in columns, I'm going to have different fitness landscapes, so different computational problems. And in rows, I'm going to have different selection schemes. And within each of those, I'm going to be plotting phenotypic diversity versus phylogenetic diversity. And so if we look at tournament selection, which is kind of the simplest one of these selection schemes, we can see there is a correlation between these. It's not the strongest correlation, but it's there. And similarly, if we look at random selection, which is just a control, that looks similar. So that this is implying like, there is shared information between them, but also, not all of the variation in one is explained by the other. Interestingly, as we start to add more complexity to our selection schemes, specifically, these are ones that maintain diversity and kind of introduce ecological interactions, this correlation starts to break down. So for these middle 3, where there's ecology going on, these R-squared values, the relationships between these variables are very low. In many cases, they're not significant. We do have one exception here, which I can talk about if people are interested. But for the most part, what we can see is, first of all, none of these R-squared values are high. So we can say pretty conclusively that phylogenetic and phenotypic diversity are not the same thing. They do tell us different information. And number 2, the exact relationship between them does depend on the specifics of the evolutionary scenario. So the fitness landscape and also the evolutionary dynamics that we have incorporated into what we're allowing evolution to do. So those are both important to keep in mind as we ask our second question, which is, does phylodiversity actually tell us something that is useful? Does it help us better understand the evolutionary process? And so this is a really hard question to ask because diversity and success, and in this case, I'm defining success as ability to continue increasing in fitness and potentially evolve novel traits or invade new ecological niches. So this relationship between diversity and success is this very tightly coupled feedback loop. I already said we need diversity to succeed at evolving higher fitness or novel traits, but also, especially if we're in a regime where we're maintaining diversity, anytime we evolve a new trait, that's going to contribute to diversity. And so it's really kind of impossible to separate loop, which has led historically to a lot of people kind of throwing up their hands and saying, hey, this algorithm makes both fitness and diversity go up, and that's the best we can do. And when we're just looking at one notion of diversity, that's not the worst thing. But if we want to start uncoupling like the effects of different types of diversity on evolutionary success, again, success is a very evolutionary computation centric term, but ability to continue increasing fitness, we need to like have a better way of thinking about this. And so one approach to this is the idea of causality analysis. So if we have two time series, so two values that are both measured over time, in this case, let's call them A and B, is there a way that we can figure out whether or not B causes A? And I know intuitively we probably all think, we have to be really careful. Correlation doesn't imply causation, and that is 100% true. However, when we have time series data, we can exploit the fact that we know stuff about how time works, basically. So there's this notion of causality called Granger causality, which is based on two main insights. So the first one is the past influences the future does not influence the past. And so what that implies is that if we have these two different time series, we can look at the amount of information that a previous state of one of the time series provides about the current state of another. And so this is an operation called lagging. So if we lag time series B here by 1, it means we are shifting it in time by 1. And now we're comparing the current value of A at time t to the value of B at time t minus 1. And if we see here that B tells us something about A, that starts to imply some sort of causality, because we know that A could have been caused by B, but B could not have been caused by A. Now there's a problem here that I don't have time to go into the details of, but there's kind of a null hypothesis here that's really important to keep in mind, which is that the variable basically causes itself. And so for reasons I'm happy to talk about later, we also want to compare A to a lagged version of itself. And really, if we want to know that B is causing A in this Granger causality sense, what we want to show is both that lagged versions of B provide information about A, and also that information is not information we already got by looking at a lagged version of A. So we need lagged B to provide information above and beyond lagged A about current A. Okay, so can this help us? So here, let's just start by looking at one of these selection schemes, tournament selection, and this is phylogenetic diversity. So on the y-axis here, what I've got is the amount of information that the lagged version of phylogenetic diversity provides about fitness above and beyond the information that the lagged version of fitness provides about fitness. So on the x-axis here, I've got the amount of lag. So for instance, this box plot, oh, and I apologize, these are all missing a 0. So this box plot here is showing the amount of information that phylogenetic diversity 1,000 time steps ago provides about fitness right now, above and beyond the information that fitness 1,000 generations ago provides. So the first thing to notice here is just these are not 0. Like there is information provided by phylogenetic diversity, even from incredibly long periods of time ago. Like I'm very impressed that phylogenetic diversity 40,000 generations ago can tell us anything about fitness now, but it can. So this implies that in this Granger causality sense, there is causation happening here. And if we look at this across all of these selection schemes, we can see for all of them, there is some level of causation happening here. It varies a lot based on the selection scheme. Note that these axis ranges are not the same, so you can see what's going on. And so lexicase selection, or sorry, fitness sharing and eco-EA are the two where we're kind of seeing the most. I guess lexicase too. Basically, these more complex selection schemes where there is more ecology going on are the ones where we are seeing stronger causal effects of phylogenetic diversity on fitness. So this is phylogenetic diversity. One of the other questions is, does this provide information that phenotypic diversity doesn't have? So let's look at what phenotypic diversity would tell us. We can see the amount of information that phenotypic diversity provides us is generally lower. Not always, but usually. Again, there seems to be relationships between the different selection schemes here. So from that, we can conclude, so like I said already, phenotypic diversity and phylogenetic diversity are different things, especially when we have ecology going on. And from this causality analysis, we can kind of preliminarily say that there is a causal relationship between diversity and fitness, and that phylodiversity seems to have a stronger causal relationship, at least in the circumstances that I tested. Importantly, that was all in one fitness landscape. In the first set of results, I showed the fitness landscape matters a ton. So this might not generalize across fitness landscapes, but it's a way that we can start to get at what is actually driving this feedback loop. So I want to thank a ton of people who have influenced my thinking on these questions, and also ICER again for letting me run lots and lots of jobs on their cluster. And I will take questions now. Thank you all for listening.
So Emily, should we raise our hands?
Yeah, let me figure out.
So I'm raising my hand.
Oh, you're raising. Go for it. Yeah.
All right, so that was a really super cool talk. I mean, 2 super cool talks in a row. On my mind is sort of discombobulated. So there's a reason I left ecology, and your talk underscores why I left ecology. But one of the points that I wanted to make was, your point about time delay, and particularly if you're doing ecology and if you have systems that are weakly nonlinear, are these time delays going to lead to unique trajectories each time the system runs? And so the predictive capacity of trying to go backwards is actually lost because of the nature of the interactions. And that's why I quit ecology.
Yeah, I definitely feel your pain there. Yeah, I mean, I think I kind of glossed over here was the fact that this is me attempting to summarize these across many runs of evolution. So those box plots there were each 50 runs for each condition. And so we're looking at the average amount of information we get within each run, but there's not a good way to summarize this across runs other than those box plots. Great. Okay. Looks like we're about at time. So I'm going to move on to the next talk, but if anyone has future questions, I will be happy to answer them later. So next up, we have Arend Hintze.
Yeah, hi. Let me share my screen. You can see this, right? Okay, I'll just start, right? So title of the talk, Evolvability of Developmental Encodings. And it actually goes back to a discussion that I had with Chris more than a decade ago, where I said, you know what, in a Vedian, well, the genotype is the phenotype. And he's like, no, the genotype isn't the phenotype. The genotype is the genotype, the program of the Vedian, and the phenotype is what the organism does, right? The computations that it performs. But I meant something else. And what I meant is this thing here. When we talk about encodings, when we talk about natural organisms, you have DNA. DNA makes proteins. Some of those proteins are transcription factors. Those transcription factors regulate each other and also proteins. And pretty much together with the proteins and all the other things that DNA does, define everything that the cell does. Well, the cell actually divides in a multicellular organism. So I'm not talking about microbes, I'm talking multicellular organisms. Well, they make more cells when they actually go through a developmental stage. And of course, proteins are there for cell-cell signaling that is used to, again, cause transcription factors to be expressed and to make cells different from each other. And all of this means that you get pattern formation and differentiation. And of course, the moment you have cells.
Just FYI, Arend, there is a thing blocking kind of the right part of your screen. It says build order.
Oh, that's funny. Let me remove my build order. Thank you.
Sorry about that.
It's gone, yeah? Okay,
yeah,
so
thanks.
Where was I? So cells form more cells and they do pattern formation and differentiation. And over time, that means that cells create the body plan and organs with it. And of course, one of those peculiar organs is the brain, and the brain makes you put feathers on your arms and you fly. I mean, works only in 50% of the cases, and Daedalus, Icarus kind of didn't. But the point is that, well, there's some kind of encoding between the genome and the actual phenotype that is much, much, much further related than in Avida, where you have code and phenotype. So if you want, translating the genotype into the phenotype, that's the encoding, okay? And of course, in biological organisms, the encoding is actually much more complicated than in our computational models, right? In our computational models, Avida, is encoded directly, right? You have the code of all the commands in a neural network, recurrent neural network in this case. You have the weights define the function. In genetic programming, all the nodes define directly the functions, and even Markov grains have this thing where you have genes, but each gene defines one gate. So all of these computational models can be described as direct encodings. They have some kind of one-to-one mapping. You have a genome, and in the genome here is 16 values, and each of these values defines one weight. And well, if you mutate one of those weights changes, right? So a direct encoding is one-to-one mapping between genotype and the final product. What it does is a different question. Okay. If you have an indirect encoding, however, well, then you don't have this one-to-one mapping. You typically have a shorter symbol sequence, and those symbols, when mutated, actually translate into larger effects. It's not that one element in the genome actually affects one element in the network. It affects many, or it can affect many. So it's a one-to-many mapping. That's called an indirect encoding. And of course, the most famous example, I think, is hypergeometric encoding using CPPNs. CPPNs are compositional pattern-producing networks. So you have a string that defines a little mini neural network. And this one has an input I and an input J, which are nothing else than the coordinates into this weight matrix. And out comes the weight for each of these elements. So when you have a mutation here, it basically means that when you go feed in each I and each J, you get the weight out of it. And if you have a mutation in the string, then typically you have a very different CPPN. And of course, the effects are much larger, or can be much larger. So that's an indirect encoding. Now, what's a developmental encoding? And a developmental encoding is trying to actually extend this notion, right? It is an indirect encoding, but the indirection actually unfolds or develops over multiple time steps. That's kind of how we define this. So let's imagine you have a genome, and it's a little bit hard to see, but it's a genome of zeros and ones or black and white pixels. And we would like to, develop or unfold this genome over time. So we have sequential updates, or AKA development process. And just to illustrate this, we apply a cellular automaton with a particular rule over it, and you see that you go from genome or many time steps to, the phenotype in the end, or whatever we use as the phenotype. So there's a translational step that goes over multiple time points. And of course, you see that the moment we mutate that particular genome, this effect actually translates over many of these updates and can have drastic or not so drastic effects down here from the end. So when you have an indirect encoding or a developmental encoding, you have much stronger effects from mutations than you would have with direct encodings. That's kind of the basic idea. And developmental encodings, at least from my point of view, takes this to the extreme, right? You have way more extreme mutations than you have indirect encodings alone. Okay. We had this workshop in 2020 at ALife, where we talked about development encodings, and we presented a couple. So let me show you the controls. First of all, in direct encoding, we want to encode a recurrent neural network, and we have our genome, and we just translate the genome directly into the weight matrix of the RNA, one-to-one, perfectly straightforward. Indirect encoding, well, we use a CPPN. So we have the genome define the CPPN define the weight matrix of the RNA. And now we come to our first developmental encoding. We use a GRN, gene regulatory network. And the gene regulatory network is specified by a C matrix, so that's the interaction matrix of all the elements interacting with each other. So we take the genome, populate the C matrix, let the developmental GRN, or the GRN run for, in this case, 200 updates, and then take the values of all these genes expressed and use them to populate the RNN weight matrix. So we have these temporal time component to all of this. There's more that you can do with these things, but for this example, let's just be it. Well, we found out that GRN is actually kind of not evolving quite well, so we played around with this some more and made this more extreme. We take our genome, translated that into a CPPN. The CPPN now defines the weight matrix of the GRN. We run the GRN, and then the GRN actually defines the weight matrix of the recurrent neural network. So two sort of, indirect and developmental step. And it turns out that this evolves quite well, at least in the experiment that we used there. Now, for good measure, we actually flipped this around. We said, well, what happens when you do this the other way around? We take the genome, define the weight matrix of the GRN, let the GRN define the CPPN, and then the CPPN actually defines the weight matrix of the RNN. Possible, worked terribly. So, okay, apparently you have to have the CPPN before the RNN for this to work. Anyways, we have now 5 different encodings, a direct one, an indirect one, and 3 developmental encodings. Now the question is, do developmental encodings evolve better? Yeah, that's a good question, but on what task? Because remember the no free lunch theory, every optimization or search algorithm is optimal for one particular problem. You can find the worst example on every task. So no search works best on all. So it's perfectly impossible to actually say that developmental encodings evolve better on all tasks. So you have to test them on a subset of tasks. Unfortunately, from 2 years ago, GPTP conference, we did the buffet method where we actually tested a particular scheme on 9 different evolving environments. There's the balancing pendulum task, there's the maze world, there's the block catchy work. Anselmo, this is the associative memory task from you, navigation task, the simple, can you perform an XOR, symbolic regression, value judgment, you have to tell which of the lights is stronger on, or a foraging task. So 9 different tasks from sort of embodied cognition and abstract things like XOR symbol regressions. So let's evolve these 5 different encodings on these 9 different tasks. So what you see here is sort of, 50 replicate runs for this combinatorial matrix of 5 by 9. It's a rather a nasty computational thing, but each of these plots shows you, I think over, it's not on the x-axis here. I think it's 50,000 generations. And on the y-axis, you have the performance, and it's averaged over 50 replicate runs. The black line is the, the red line is the average, and the black line is the noise around it. Okay, point is, once you run all of this, well, direct encoding actually beats all the other ones, right? Direct encoding is always better. That's already funny because other people claim that indirect encodings are better than direct encodings, but on these 9 tasks, it's not. In fact, the indirect encoding ones, the hyper encoded ones, are actually second place on 1, 2, 3, 4, 5, 6, 7 of those tasks. And only the GRN encoding and the CPP and GR encoding are actually second place on 2 tasks. The CPP and GRN, the one that we found to work really well, is best, or second best on value judgment. And GRN only is the second best on Adlon maze compared to hyper encoding. So yeah. Developmental encodings aren't better. At least that's what we find from this encoding, from this experiment. However, there's a caveat. And the caveat is that these are all static environments. None of these environments changes over time. And of course, we know that organisms, who all more or less have a developmental encoding phase, actually evolve in variable, changing environments. So maybe we're testing them on the wrong substrate. So let's test them on the dynamic foraging environment. Dynamic foraging environment is really simple. You have blocks of food cubes in there. You have to forage these food cubes, and the order in which you eat the food cubes determine the payoff that you get. So for example, if you eat a red food cube, after you eat a red food cube, punishment, that's really bad for you. If you, however, eat an red food cube after an orange food cube, well, there's a really good score. So you have to figure out in which order to eat these food cubes. And once you do this, well, you have to evolve to this, but we keep changing the values of these food cubes up and down using a sine wave function. So this dynamic foraging environment changes constantly, and we can control how fast it changes. So let's evolve our 5 encodings into these environments. And cool, GRN encoding seems to work better than the direct encoding, with one problem. When the speed is 0 and it's a static environment, it's already better. So in this particular environment, GRN encoding actually works better than direct encoding. However, this is the cool part, our GRN CPP encoding works worse in the static environment, but works better in most of the other speedier changing environments. Unfortunately, there's a little bit of low statistics up here, so we're still running more experiments. Anyways, let's come to the summary. So no, not all developmental encoders are created equally, right? We created one which completely doesn't work and one that works quite nicely. They appear at best to be as good as direct encodings, at least on some static tasks. I mean, the most we can test. Well, they're occasionally better than indirect or hyperencodings on some static tasks, but in a changing environment, they can perform better depending on the speed of the change. So what's the hypothesis from here? Well, we already said that developmental encodings have much larger mutation effects. So the mutation effect size distribution apparently matters for the rate of adaptation, given the different environments. That's one thing. And the second thing is changing environments might have a very different optimal mutational effect size distribution or require a very different mutational effect size distribution than static ones, because they keep changing, and that's a very different challenge for evolution than it's a static environment. Okay, thank you very much. Of course, lots of high performance computing done by Isa and Balakiran Mantri and Kiran Sai Tenaru, who doesn't want to be photographed, who did actually all the computational work. Thank you so much.
Great talk, Arend.
Thank you.
Got plenty of time for questions. Fantastic job keeping it to 12 minutes.
You want me, so. Yeah.
So yeah, to ask questions, feel free to either do the raise hand button or post them in chat. Okay, I see there's a raised hand. Wait, who is, oh, Una May, go for it.
Hi, everyone. Let me see if I can turn on my video too. And I'm not sure I have a clear question. I'm really struck by the fact that you almost have like a different evolutionary rate with your mutation effect in these two different models that you've put in sequence. And I'm interested in whether one could actually compare the speed at which one adapts to the other from the encoding, not from the encoding, but yeah, in a sense it is what you're using a rule, right? And you're mutating it. Because I'm just wondering whether it's about like almost matching the impedance of the environment, right? So impedance matching the environment. So the environment has a certain amount of dynamics as it's shifting. And you've got two components and they're both capable of having different mutation effects. And you really want all those three things to sort of couple together, right? And tune together. And so do you have any sense of like whether one's faster than the other or slower than the other? You can measure that, right? By the way that you've done the encodings?
Yeah, actually, I think this is a brilliant question because the first thing we wanted to look at is the actual mutational effect size distribution. So what is happening when mutations happen? How do they change? What's the difference that occurs? And I mean, only from a sort of, brief look, we actually visualized the matrices and looked at the matrices over time. And it turns out that, of course, if you have this sort of second layer encoding where you look at the GRN change matrix given the CPPN, or even the third one, right? The further down the line you are, the more drastic the changes are due to mutations, which, I mean, you kind of expect, but, it's also nice to see this. And I think it's an interesting point. I mean,
you could even put all three in a row, right? You could use the same encoding and change a parameter of it, and then you get a sense of which one had more drastic effects, and how you can modulate the effects of each three. Like it's almost like I'm not sure whether it's important that you had a GRN and then a CPP, or whether you had just two GRNs, but they differed parametrically.
Yeah. I mean, this is a comment that we had in the discussion, because we can sort of arbitrarily nest these things behind each other, right? And if you think about biological organisms, I mean, we basically came up with like 9 layers, right? It's like cells to cells, or sort of proteins to proteins, proteins to cells, cells to cells, cells among these other tissues, blah, 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 blah. And we're using 3 layers, right? So we're still not at the point. So maybe, and that's, I think, the easier experiment is to basically just keep adding layers, right? At what point does this stop making sense? Or maybe it just evolves and we're sort of, fuzzing around about something that we don't need to be afraid of. It's just whatever happens. But yeah, I like the
the other thing is if you could evolve the rates of those mutations, not the mutations, the effect of, not the impact of the mutations, but the mutation rate on the system, then the point is that like the complexity of the environment, it's almost like there's having thought about this a lot, and you can tell me whether my intuition is completely wrong. It feels like the complexity of the environment very much matters. And you went from static to dynamic, but I think there's a whole series or a whole taxonomy of complex environments. And the complexity of the environment, in a sense, you want it to be entrained, or you want it to entrain, you want the parameterization of these different layers of your network to entrain to the dynamics of the environment. And you could do that with something like an evolving parameter. The problem is that you have to have a fairly complex environment, or you're just going to see these very short-term effects that are essentially artifacts.
So this is a fantastic conversation, and I hate to cut it off, but we have to move on to the next speaker to make sure we have time. Super interested to hear Arend's thoughts on that in the future. Thank you again, Arend. Next up, we have Stephen Kelly.
Sorry, I was muted. You can all see the slide there. Okay, great. So my name's Stephen. I'm going to be talking about evolving hierarchical memory prediction machines in multitask reinforcement learning. I want to thank my co-authors there as well. So RL in a nutshell is a type of machine learning in which the agent learns its behavior through trial and error interaction with the environment. So the goal is for the agent to learn how to map states or situations to actions such that reward is maximized. And interactions are episodic. So the environment, an episode starts in a specific spot, and an interaction continues until the agent either succeeds or failure, or a time constraint is reached. So in evolutionary RL, fitness might be the average reward over, say, 20 episodes. In this work, we're going to be looking at the set of classic control problems that are included with OpenAI Gym. So these are well-known reinforcement learning benchmarks. For example, the cart pull task, in which the agent has to observe the state of the system by means of, in terms of, it's like a cart that can slide horizontally on a frictionless track, and there's an inverted pendulum. So it gets the X position of the cart and the X velocity of the cart, and it gets the angle of the pull and the angular velocity of the pull, and it needs to apply a bang-bang force in order to balance the pull. Other tasks, such as pendulum, are continuous control problems. So the agent needs to apply a continuous torque value to the pendulum joint in order to swing the pendulum up and then balance it. So the goal of multitask reinforcement learning is to develop a single agent capable of operating in multiple environments. Some of the challenges are, for example, the distraction dilemma. So the magnitude of the rewards for different tasks, and I'm just going to turn on my video here, may be different. So certain tasks may appear more salient to the agents. We have catastrophic forgetting. So the agent needs to figure out how to avoid unlearning behaviors that are only intermittently important over time. And negative transfer. So jointly learning dissimilar tasks might be especially difficult. And scalability. So jointly learning n tasks should not require the agent be n times as complex. Plus, in classic control problems, as we're looking at them, as I mentioned, we have a mix of continuous and discrete valued action spaces.
Stephen, just FYI, you have a thing blocking the middle of your screen.
Yeah, I was trying to, my computer's being a little sketchy, so I was trying to troubleshoot there, but it's good to know you're still there. Okay, so we have a mix of discrete and continuous actions, and the environments are only partially observable. So I'll talk a little bit more now by describing the agent environment interface that we're working with. So the agent has only 2 inputs, but all of these control problems have between 2 and 4 state variables. So the agent is actually blind to any of the velocity information of any of the tasks. And notice the agent has no input that would tell it what environment it's interacting with. Agent has 2 outputs, one for discrete actions, which can take one of 3 values, and one for a single continuous action. So the environment will interpret those actions differently depending on actually which problem the agent is interacting with. So the goal, the agent needs to learn how to recognize the different tasks just by observing how the state variables change over time, and then needs to define an effective control policy. So the program representation we're working with starts with two memory vectors. One, M, is stateful, so it's only reset at the beginning of each episode. And the second one is stateless, it's reset prior to each program execution. So that M register allows an agent to integrate information over time so that it can combine variables to hopefully predict those unobservable parameters. This is what the program representation looks like. It's a pretty standard linear GP register machine. The input is the state of the system. And then there's a series of instructions that operate on those two memory registers as well as the state. And there's two outputs, which are one discrete action and a continuous value, which is whatever value is stored in memory register 0. It also returns the value of the stateless register 0. And that's essentially like a bid value, because in the hierarchical system, multiple programs will collaborate with each other. So a single program can be seen as sort of defining a context for one action, or for the two outputs given the current state of the environment. So in order to introduce hierarchies, we first introduce teams. So a team represents a complete decision-making policy by indexing some number of programs from a different population. At any given time step, the program with the highest bid value gets to run its actions or its pair of actions. So initial populations start off like this, and then over time, variation operators are allowed to manipulate these pointers such that teams, rather than executing their pair of actions, they can defer decision-making to another team in the current population. So they can essentially now, at this point, T3 is learning a context in which to deploy a previously independent policy. So we get this kind of compositional evolution in which hierarchical structures can emerge that sort of recombine multiple agents that were once independent. Now, in order to judge their multitask capability, we formulate a multitask fitness function, which is essentially just normalizing each agent's fitness score for each task relative to the current population. So we get the population-wide min and max scores for a task and normalize the agent score based on that. And then their overall fitness is the minimum of their normalized scores over all of the tasks. So the point is that the agents are judged by their weakest link, the task that they perform weakest on. And then the GA looks like this. So we initialize 1,000 teams with 2 to 10 programs each, evaluate those teams in 20 episodes. And then we keep elites. We keep one niche for each set in the task power set and delete the remainder. So for 6 tasks, there's 63 different sets. And for each set, we keep 25 of the elite agents. And then from that, we sample, clone, mutate, and apply crossover and start again. So this is sort of an important thing I'll come back to at the end. It's sort of like the strength and the weakness of the system. And this is the result. So this is a single experiment. And what we're seeing here is the evolutionary progress relative to all 6 tasks. So the dotted line represents the fitness of the single best policy that is best at one problem. So these are task-specific single task agents in the dotted line. And the solid lines represent multitask agents. So for example, the black solid line is the fitness of the single best multitask agent. So it's the same agent performing at that level in all of these problems. And this is what that best multitask program graph looks like. So in this hierarchy is shown just the teams, and we can analyze it by replaying it in all 6 tasks and seeing which parts of the hierarchy contribute to decision-making while it experiences what problem. So of course, the decision-making always starts at the root node. So the root contributes equally to every task because it contributes to every decision. But then as you go down the hierarchy, you can kind of see that things start to specialize. And we can analyze things like, of course, it makes complete sense that there's 2 different versions of mountain car. One is a continuous control problem and the other is a discrete control problem. So it makes total sense that teens tend to sort of specialize or generalize over those two problems. Some of the other combinations are less intuitive. And we can look at the runtime complexity in terms of the number of instructions that are executed per decision. And of course, it's different for every problem, but it's, you know, never any more than about 300. And I compare that with OpenAI has a leaderboard for what is the best agent developed for each problem. And this deep learning D4PG agent is the best on most of them. And that's a deep neural network with at least 120,000 weight parameters. So the complexity is significantly more than the programs that we're able to evolve. Plus, this deep learning agent is, it's a single task agent that's evolved from scratch for each problem. It can't do multitask. We can also look at whether these agents actually are able to make a prediction of the unobservable states in memory by analyzing their memory registers and see if we can find those unobservable state variables. And so this is an example for the pendulum task where you can see the agent's prediction of the velocity is very accurate up to the first about 100 time steps, and then it sort of dips off a little bit. And that's makes sense because as the agent's trying to swing up this pendulum, it needs some kind of prediction of the velocity to even know which direction to apply force to. So that prediction of the unobservable state is really important. But once it gets vertical, it's less important. So I'm just going to play. This is a video of what it looks like in action. So you can kind of get a sense of the degree of path switching within the hierarchy. Of course, only part of the hierarchy is actually used for the pendulum task. So the rest is sort of grayed out at this point. So a quick, flashback to some of those challenges. We were able to build a single task or a multitask agent that could operate in multiple environments. The distraction dilemma was taken care of by normalizing the rewards, so pretty simple. Catastrophic forgetting, I think, is managed by having a niche for each task set and then building agents with a hierarchical recombination from those niches. Negative transfer is minimized again through hierarchical task decomposition. So that allows the multitask agent to subsume specialized behavior and switch between them rather than having to form like a totally generalist policy. It can sort of decompose the task like that, and it can use that to also exploit positive transfer. Scalability is addressed here by starting simple programs and complexifying through environmental interaction, but there's a huge caveat there. Partial observability, again, we can deal with that by keeping some registers stateful. And I really just have linear GP to thank for that because it's very easy to do with that system. And same with managing discrete and continuous action spaces. We just evolve programs that have two outputs. Search space is slightly larger, but it's no problem. And that's it for me.
Thank you, Stephen. Great talk. We've got some time for questions. Good job staying on time. So yeah, feel free to ask them in the chat or to raise your hand. I guess while other people are thinking, I've got a question.
Sure.
So I know you've been looking at kind of like the complexification of these and kind of the size as a mechanism for that. Do you have a sense of whether, I know sometimes when people have studied complexification in GP, they've looked at the extent to which the complexification is limited to like just as much as you need versus kind of like just ratcheting up constantly in complexity. Have you thought about that at all with these? Like, do you think they're minimally complex, or are they getting more complex than they need to?
They're probably getting a bit more complex than they need to. The way that I deal with that in principle is by starting simple. And then I didn't mention, but also all of the variation operators are biased towards simplicity. So it's more probable that they'll get simpler than more complex. So I can sort of reasonably say that the complexifications are due to an increase in fitness.
Awesome, thanks. Looks like we have a question from Risto.
Yeah, usually when you do multitasking and supervised learning, you actually get a benefit that your individual tasks do better when you're also learning from other tasks. And here it's not happening, obviously, because if one single agent tries to do multiple tasks, but is there some way that you could use the knowledge that you learn from multiple tasks to do better in every one of them?
That's a good question. I think it's definitely possible. And again, I would approach it through the hierarchy. So you could, recombine multiple different specialists that solve the problem in different ways in order to make a more robust policy. But I think the thing that makes this specifically challenging is the fact that it's partially observable. So I didn't sort of get to the point. I viewed the multitask learning as more of a challenge than a potential thing to leverage in the evolution.
Awesome. I see we have a question from Una May, but unfortunately we are at time. So hopefully you can chat about it offline. Next up, we have Risto Miikkulainen.
Yes, just a sec, I'll share my screen. Okay. Yeah, so yes, so this is a talk about some research we've done at UT Austin with Cem Tutum and Suhaib Abdulquddus. Cem, of course, is a longtime Beacon member, and Suhaib did his master's thesis part of this work. This is funded by DARPA's lifelong learning problem, and I think this is very relevant to that problem, lifelong learning, but not quite doing that yet, but instead trying to do as much extrapolation and generality as possible to serve as a foundation for lifelong learning. So the generalization is the main question. That if you have these agents and you try to deploy them, the real world always gives you surprises. It's impossible to train with all kinds of possible situations, and you would have to have generalization ability that actually makes you perform well, even if the world is not what you expect. And we can see that in many different domains, autonomous driving, robotics, process control, healthcare, finance, there are always surprises. So, and if you don't tackle this problem, you can get these catastrophic errors and outcomes that then really ruin your day. So our solution that we explore here in this work is that maybe we could separate the modeling of context from the actual performance, from the actions that you perform in the task. And this way, if you learn something about the context, you could make the context look more similar so that you could still do something even if you have a surprising environment. And this originates from the insight that Shun Lee had a few years ago on poker, a different domain, where he was trying to model the opponent. You have different kinds of very aggressive opponents, or passive aggressive, all kinds of different opponents that prefer certain kinds of strategies versus others. And if you're playing many hands against them, you learn what their playing style is like, and then you can customize your own strategy against those opponents. And this actually worked really well in poker. It was surprising how little the learning needed to see of different kinds of opponents, and it would still scale to very complex, very powerful opponents because we were explicitly modeling the opponent. And here the idea is to take that idea and generalize it, make it more general about skills and contexts. So the context in poker was the opponent, but we can imagine different environmental contexts where conditions change. For driving, it might be more slippery, there might be raining, you might lose a wheel, you might be towing a trailer, and there are similar modifications for these other domains as well. So, and in order to test this idea more generally, we tried 3 different domains, and they are shown here: Flappy Bird, lunar lander, and autonomous driving. Before we get to those, though, here's what the approach looks like. So there are 3 neural networks, and they are all evolved. So this is a neuroevolution approach. They have different architectures. So the context integrates over time. That's how you know what your context is like. You observe the interactions with the environment, what effect your actions have on the environment over time. And then you know what the, I don't know, gravity constant is, or how much wind there is, or whatever the environmental settings are. And then you do have the skill module that looks at the current situation and makes a decision. And the controller's job is to take the suggestion of the skill and the understanding of the context and produce something that is a modulated action. You know, your current action adjusted to the current context. That's a general idea. But of course, these are modules, and you start with random weights and then you evolve it. So they take on these functions through evolution, but they do have different architectures, and you do have different time scales. And that's what drives them to different kind of behaviors, these 2 modules. Now, in order to test and see what these actually do and how valuable this general architecture is, we'll try some ablations. We leave out the skill and only look at the time history, or we leave out the time history and just react to what currently is in front of you. And here we also have to balance the number of parameters, because we are leaving out components. But it turns out that doesn't really matter. So we can really just leave out the module and look at the rest of the network as it is and compare. So this is the first domain. We call it Flappy Ball because it's not quite the same as Flappy Bird. There's more happening here. In Flappy Bird, all you can do is just flap at certain times, and that moves the bird up. And then you have to try to clear those hurdles, those openings in the pipes. But here we also give it an opportunity to flap forward. So you can adjust your speed, how fast you go. And then you have gravity pulling down and drag slowing you down. So this is a more complex domain, and it allows for more variation. So we can change how much acceleration there is as a result of each of those actions. And this way you can modify environments and see how well you generalize. There are 2 objectives. There's always a safety and performance objective. Safety is don't hit those pipes or ceiling or ground. And the performance is fly as fast as possible through those pipes. And the agent gets to see where it is in the environment and what is coming up. The second domain is the lunar lander from OpenAI GEM. Again, there are state variables that tell you where you are and whether you're rotating or not, or whether you're on the ground. And then you have your actions, which are the thrusters. There's a main thruster to slow you down, and then 2 thrusters on the sides to orient you. Safe landing means in the bottom here between the flags. And performance is try to do this fast. And again, variations include how much mass there is to the lander and how strong your thrusters are, and how much of an effect they have on the environment. The last one is Carla, and that's an automated driving simulator. Here, again, we have state values, which are the distances that you can see here to the sides of the road or the lane. And then you can steer and you can apply throttle, go faster or slower. Safety objective is to stay in your lane and wobble as little as possible. They are just simply summed up. That's one objective. And then performance is to drive fast again. And you can change again how much of an effect this turning the steering wheel has, how much power you have on your throttle. And also you can change to different tracks. And that's how you can create variation. All right, so now to train, we start by exposing the agent to different variations, but the variations are not that big. You only change those parameters that I mentioned, 10%, 20%, 15%. You do create a little bit of visibility what can change, so the agent is prepared for that, but it's very limited. And then we are evolving, as I said, these neural networks, 2 objectives, so use NHGA 2. And then we stop when we get acceptable level of safety with some minimal performance criteria. So you can't just stay still, that's not acceptable. You have to move at least, even though you wouldn't hit any obstacles if you stayed still. It's kind of a minimal criterion performance, but we stop as soon as we have a competent agent, doesn't hit obstacles in case of Carla, of course, lands safely in case of lunar lander, and flies through the track in case of the Flappy Bird. And here are some results. This is the Flappy Bird first. And now we are looking at generalization. So we are creating much more variety in the domains. So it's 75% of variation, plus or minus, in each of those parameters and all at once. So it creates a lot more variation. So these tasks are quite different from anything that's been seen during training. And then we test many different such variations, 10,000 such variations, and evaluate how well we do in those two objectives. And here is a comparison of the context plus skill architecture compared to just having a skill, no context at all. So we subtract the objective values, subtract S from CS, from context skill, and therefore we see that if most of these 10,000 tasks land somewhere on the left of the zero line, then the context skill is doing better. And here we have a maximization objective, so context skill is doing better if you're on the right side of the zero line. So as you can see, the context skill is quite significantly better than skill only and significantly better than context only. And the context and skill are roughly the same if you only have one of those. Now, to illustrate what this actually means, this generalization, it's not just your, garden variety generalization where you interpolate between your examples. That wouldn't get you very far. The training tasks are actually located inside this plus. Remember, we changed just one variable at a parameter at a time. So we are increasing forward thrust, for instance, or decreasing it, or in another domain, we might be decreasing the flap effect or increasing it. But here is what we are showing it during the testing, the generalization. All these combinations, much wider range. So these are quite different environments from anything seen in training. And here we have one plot for each pair of these parameter changes. And you can see that the light color indicates that we are doing better than, context skill is doing better than skill. So just zooming in to the generalization a little bit in one case of Flappy Bird to show you that this is actually really challenging. And we are going out and saying that this is not just generalization, it's actually extrapolation. So we are not interpolating between tasks that you're actually extrapolating far outside of it. All right, similar results with the other two domains. This is the lunar lander. And again, CS is much better reliably than pure skill or pure context. In this case, interesting enough, the skill is better than context. And that's probably because lunar lander requires certain reactivity, and that's what skill can do. And in Carla, again, CS is better than either one of those, although the differences here are not so clear. They're roughly similar in safety, a little bit better in distance, context skill is. And here the opposite is true from lunar lander. Context is important in Carla. You have to understand your momentum and inertia and in order to do well. So these are a little bit different tasks, and it's nice to see that the context skill consistently performs so well compared to the others. Now, trying to understand what actually is being learned, making it concrete. Here's context skill, and it has pretty good job at slowing down when there's a difficult challenge, like right there. There's a pipe that goes all the way down, and you have to be careful, otherwise you run into the pipe because you are too high. And this is very high, requires a lot of generalization, and context skill can do it, but the other ones cannot. They don't understand that you have to slow down enough. Like this one hits the pipe way up here because there's no way to come down. It cannot separate the different actions and slow down, take into account slowing down, and when you have to do it. Same thing here with the skill. It doesn't use the actions well enough when the action effects have changed, so that you can no longer trust just the gravity to pull you down fast enough. Now, last slide here. We said that it's the modulation that context provides the actions that works here. That you somehow understand what the context is, and then controller combines them and changes the actions. Now, we were expecting, if you really look at like what the actions are, that we would see a lot more variety. The context would provide you more differences, and you would have more wide representations. But just the opposite turned out to be true. And it's always great when something like that happens, unexpected. So here we are plotting PCA, 2 dimensions, the output actions of the context and skill modules in 2D, and we subtract the nominal case from an extrapolation case. And we're trying to see, therefore, what are the representations that context and skill provide? And are they different in extrapolation versus nominal case? And it turns out that with the context and skill, there's very little difference. Somehow, the context, evolving to get the context and skill, manages to standardize the representation so that even though the inputs, even though the environments are very different, what the decision maker gets to see from context and skill doesn't vary as much. Versus if you just have skill, just have context, there's a very different, the new environments cause those to differ widely, and it's more difficult to generalize. So that's the core. Learning context and skill in separate modules allows learning that makes it easier for the decision maker to generalize. So that's it. That's the message, that when we are explicitly representing different time scales, we get context and we get skill, and therefore we get a system that can generalize, not just to interpolate, but also to extrapolate. And in that sense, we get a controller and decision making that works in unseen situations. And that's the foundation for lifelong learning, that if something changes, you can perform something, you don't crash immediately, and then you can learn more during your lifetime while maintaining this kind of robustness. And that's where the research is going next. We'll use this as a starting point and add on more learning and try to utilize this ability to generalize even to very wildly different situations. Thank you.
Thank you, Risto. Fantastic talk. We've got time for maybe one question.
Well, one quick question. Obviously, the next set of tasks are going to involve for your autonomous driving algorithm being on the road with other vehicles, correct?
Yeah, yes, and different kinds of hazards, absolutely. And we've done that actually in the past, not in this Carla simulator, but a simulator called RARS. You can place other cars there. You do have to evolve with them. I mean, you do have to see that there are obstacles, but you don't have to recognize what they are. You just have to learn to avoid them, whether they move, whatever their behavior is. That turned out to be relatively surprisingly easy to do in RARS. And we're hoping that that might happen in Carla as well.
Awesome. I think we should move on to the next speaker. Thank you so much. This is really cool work. Next up, we have Kate Skocelas, who submitted her video pre-recorded, although I believe she and her co-authors are here and can answer questions afterwards. So I'm going to share her video, which I had a chance to look ahead at, and it is gorgeous. Okay, so here we go.
Hi, my name is Katherine Skocelas, and I'm excited to talk to you today about my project, The Evolution of Cellular Restraint in Multicellular Organisms, completed with my collaborators, Austin J. Ferguson, Clifford Boehm, Catherine Perry, Rosemary Adagi, and Charles Ofria. As you likely know, the major evolutionary transition to multicellularity shifted the unit of selection from individual cells to multicellular organisms. Post-transition, an individual cell has 2 options. It can reproduce as quickly as possible or restrain itself to work with the other cells. Restraint is a major aspect of cancer. Growing cancer cells, shown here in purple, replicate without restraint to increase their own fitness, usually to the detriment of the multicellular organism. This is why we wanted to study the pressures on low-level individuals restraining themselves for the good of the high-level individual. This is a standard multi-level selection problem, where there is a low-level pressure to increase individual cells' fitness by reproducing it as much as it can without restraint, and a conflicting high-level pressure on cells to work with their kin by reproducing with restraint, because unregulated cell growth is usually maladaptive for multicellular organisms. It is known that there is a bias towards the high-level individual when the life of the low-level individual is tied to it. However, many factors influence this, and while the restraint behavior is binary, the pressures that influence it are not. By looking at how much restraint evolves, we can determine how strong the high level pressure is. We measure restraint level as the number of mutational hits needed to revert a cell to unrestrained replication behavior. We hypothesize that organism fitness reductions due to spatial competition between cells can be sufficient to select for restrained behavior in cells, and that this selective pressure would increase in larger organisms due to the greater need for restraint. To test our hypothesis, we studied populations of evolving multicellular organisms in a system that we're calling Primordium. In Primordium, cells compete within 2D multicellular organisms represented here by the blue square. Each organism begins as a single somatic cell near the middle of a square grid. Each cell has a replication timer plus uniformly distributed noise that starts when the cell is born. When the timer elapses, the cell randomly chooses an offspring position from its 8 neighboring positions. If the cell is restrained, it only replicates if the selected position is empty. As in nature, cells are subject to somatic mutations with each replication. If it is unrestrained, the cell replicates into the position whether or not it is occupied, potentially overwriting another cell and eliminating any progress it had made. An organism reproduces when its entire grid fills with cells. As such, the speed of filling the grid is the organism's fitness. Having more restrained cells increases an organism's fitness by increasing the speed at which it fills and thus reproduces. Here, you can see the organism of white restrained cells takes less time to fill than the organism of red unrestrained cells. This is important because the population of organisms is kept at a constant size, with new offspring replacing a random organism from the population. Therefore, organisms that reproduce faster have a higher chance of producing offspring before being overwritten. For example, here you can see a population of multicellular organisms with 2 dominant lines, pink and blue. The multicellular organism in the center is full. It selects a random position and replicates into it, seeding the new cell with the sequestered germline, which also has a chance of mutation with each replication. However, evolution within an organism favors unrestraint, as you can see by the unrestrained red cells overtaking the restrained white cells, hence the multi-level selection. An organism's restraint value is the restraint of its sequestered germ cell. As you can see, having a more restrained germ cell allows the organism to accumulate more mutations before unrestrained somatic cells arise and take over. Because every multicellular organism is a 2D square grid, we can describe them by their height and width. For example, we would call this a size 3 by 3 organism. We found 3 key findings. First, we found that longer genomes reduce mutational pressure. To disentangle the effect of genome length on evolution at the population level from its effects on restraint decay at the cellular level, we built a control that artificially split the germ genome length from that of the somatic cells. This allowed us to test a range of genome lengths for the germ cells while always evaluating the organism using somatic cells with a length 400 genome. As you can see, we found that across every organism size, denoted in the gray labels, when genome lengths only affect mutation pressures at the population level, longer genomes consistently allow for larger restraint buffers to evolve. The restraint buffers for the smallest organisms level out once they can evolve to completely avoid unrestrained cells arising, but larger organisms monotonically increase restraint with genome length. The range of evolved restraint buffers also increases with genome length, because all genome lengths have results that extend to the median of the genome, which is at a lower restraint value in larger genomes. Our second key finding was that selective pressure for restraint has diminishing returns. Here we examined the fitness benefit of 1 additional bit of restraint for different organism sizes and restraint levels. While increases in restraint are always beneficial, we observed a diminishing return in that benefit in practice. Small organisms see the greatest fitness boost just above the restraint threshold, after which the fitness advantage for additional restraint quickly diminishes once an organism's restraint buffer is high enough that unrestrained cells never appear during its lifetime. Larger organisms have a smaller initial spike, but given that they experience more cell divisions in their lifetime, this saturation point occurs at larger restraint buffer values. Thus, selective pressure persists to much higher restraint levels in larger organisms. Finally, our third key finding was that when all other factors are controlled for, larger organisms evolve greater restraint. When we removed evolutionary noise by simulating an infinite population, and we evened out selective pressures by simulating an infinitely long genome, we were finally able to see a monotonic increase between restraint and organism size. Here we showed the evolved restraint buffer in the infinite population model, first for the default 100-bit genome, and then for the infinite genome. Since this combination of controls changes the trend, we conclude that the downturn pattern must be a combination of decreasing selective pressure and increasing mutational pressure as restraint buffers increase. In nature, population sizes are limited, but are often much larger than the size 2000 populations that we tested. Similarly, biological genomes are not infinite, but are typically significantly larger than modeled here. Thus, our system's infinite population and genome model highlights factors that could exist in nature. In this study, we have shown evidence that the pressure imbued by the management of space as a resource can contribute to the evolution of increased restraint in larger multicellular organisms. If early multicellular organisms were selected to be more mutations away from an unrestrained state, this could set up the evolution of more complex forms of coordination and even development. In future work, we look forward to building out more realism and complexity in Primordium, addressing additional issues relating to the transition to multicellularity.
Great talk, Kate. That was super interesting. We've got plenty of time for questions, so everyone feel free.
People can also ask questions later and we can get back on time. That's fine.
That is true. Yeah, I guess we are officially at the end for this session. Awesome. Yeah, I guess we can officially end here. That was a fascinating talk. Yeah, thank you so much to everyone who participated in this session. Lots of really interesting talks, good discussion. Great to see all of you.